So, today we're diving into one of the most incredible processes in the entire universe. We're talking about how a simple string of molecules gets the instructions to fold itself into a complex, life giving machine. It's absolutely fascinating.
So let me ask you a question. What's the one thing that connects the power in your muscles, the immune cells fighting off infections, and even the oxygen that you're breathing right now? They seem totally unrelated, right? But they all share this deep, fundamental biological secret.
Well, the answer is proteins. And it's not just the proteins themselves, but specifically how they fold. You can think of it like microscopic origami. You start with this long, flat chain of molecules, and it gets folded into a very precise, very functional 3D shape. And it's that shape that gives each protein its unique job.
So let's kick things off by talking about why we call these things master molecules. And really, it's simple. Life as we know it would be completely impossible without them. Let's just look at some of the different hats these proteins wear. I mean, the sheer variety of jobs they do is just incredible. They give our cells their structure, they act as messengers like hormones, they power every single muscle movement, and they're the frontline soldiers of our immune system. And the key thing to remember, the big takeaway here, is that a protein's specific shape is what determines its specific job.
Okay, so if proteins are this incredible finished origami, then what's the paper they're made from? Well, let's meet their fundamental building blocks, the amino acids. Now, every amino acid has kind of the same basic setup, the same core structure, but the real magic, that's in what's called the R group. This is the one part that's different for each one, and it gives it all these unique properties, things like its size, its electric charge, and how it feels about water. It's the R group that makes the whole protein alphabet so incredibly versatile. So how many of these letters are there in this alphabet? Just 21, that's it. From just these 21 amino acids, our bodies can build this huge, unbelievably complex library of proteins, each one with its own specific sequence and its own purpose. It's a ridiculously efficient system. But here's the catch. Our bodies can't actually make all of them from scratch. We can whip up 12 of them, and we call those the non-essential amino acids. But the other 9, the essential ones, we have to get them from the food we eat. This is a perfect example of how our biology is directly hardwired to our diet. All right, we've got our building blocks, the amino acids. So how do they go from just a simple string into a fully functioning machine? Well, this amazing transformation happens in 4 distinct levels of folding. You can really think of this as a roadmap. We start with the basic sequence of letters, then we add some local twists and folds, then we bundle the whole thing up into a compact 3D shape, and sometimes we even combine it with other folded chains to make a super protein. Let's break down each step. First step is the primary structure, and this is actually pretty simple. It's just the specific order, the sequence, of amino acids, all linked together by really strong bonds. It's literally like spelling a word. The sequence is everything. And as we're going to see in a minute, getting even one letter wrong can change the entire meaning of the protein. Next up, we have the secondary structure. This is where that long chain starts to form these cool, localized repeating patterns. The 2 most common are the alpha helix, which is a tight coil, kind of like a spring, and the beta pleated sheet, which is much flatter, more like a folded fan. And both of these shapes are held in place by much weaker hydrogen bonds. And this brings us to the next level of complexity, the tertiary structure. This is where all those helices and sheets and all the other bits and pieces of the chain fold up on themselves to create this final, unique, and often kind of globular 3D shape. For a whole lot of proteins, this is it. This is their final functional form. So what holds this super intricate 3D shape together? Well, it's all about how those different R groups interact with each other. It's like a whole toolkit of different bonds comes into play. You have some parts that hate water, so they tuck themselves away on the inside. You have strong bonds that act like little safety pins, locking things in place. And if you disrupt these bonds, say with a lot of heat, the protein unfolds and completely loses its function. We call that denaturation. The perfect example? Cooking an egg. The heat permanently messes up the protein structure of the egg white. But wait, there's more. For the really big jobs, some proteins have a final level, the quaternary structure. This is when multiple, already folded polypeptide chains, we call them subunits, all come together to form one giant, fully functional complex. The classic example you always hear about is hemoglobin, which uses 4 separate chains working together to carry oxygen through your blood. So, we've seen how it's supposed to work.
It's a beautiful, elegant process. But what happens when there's a typo in the instruction manual? Let's look at a really powerful real-world example: sickle cell disease. The whole problem comes down to a change on the tiniest possible scale. We're not talking about a messed up chapter or even a wrong word. We are talking about one single letter, just one amino acid in a chain of hundreds. And that one tiny, tiny change, well, it changes everything. It's just a stunning illustration of how absolutely critical that primary sequence is for the final protein. So here's exactly what happens. In the primary structure of a part of your hemoglobin, a single amino acid called glutamic acid gets swapped out for another one called valine. This one little substitution completely changes the properties of that spot on the protein, and it alters how the entire thing folds. This misfolding then causes the hemoglobin molecules to start clumping together, which deforms the red blood cells into that classic sickle shape that gives the disease its name. Understanding this at a molecular level is exactly why we can now develop therapies, like gene editing, that aim to go in and correct that one specific typo at its source. It's incredible. So as we bring all this together, it really boils down to one simple yet incredibly powerful idea. In the world of proteins, shape is absolutely everything. If there's just one thing you walk away with today, please make it this. For proteins, structure dictates function. The sequence determines the structure, and the structure determines the job. It is the central idea in all of protein biology. And that leaves us with a pretty mind-blowing final thought. We now understand this incredible process of microscopic origami. So, what's next? If proteins are literally the blueprints and the machines of life, what could we design? What diseases could we cure? What could we build if we truly master how to write this code ourselves?